Hello, these are flipped notes 2-4, and we are learning about how the brain is studied. My Bitmoji is getting punched in the head, maybe getting a traumatic brain injury or a TBI, because that is actually one way that we study the brain by looking at people who have been injured and then comparing what they were like both before and after their accident. So we can learn a lot from brain injuries. Studying brain injuries like that would count as a clinical observation, especially if we know information about what the person was like beforehand. We can observe the effects of brain disease and injuries. In class, we'll talk about Phineas Gage, who is probably the most famous example of a clinical brain injury case study, in which he survived major trauma of a rod shooting through his head. Another way to study the brain is to actually manipulate it, meaning to do things like stimulate a specific part of the brain, either chemically stimulating it or magnetically. An electrode could be pushed or inserted into the brain. In this case, this rat has the ability to press a lever, and every time it does that, it stimulates the electrode in his brain. We'll talk about an experiment it's actually a little bit later in the chapter, but where a rat had the ability to stimulate its own hypothalamus over and over again, and it did a lot. We'll talk about that more why in class. And then another way to study the brain is to look at EEGs to record the electrical activity or the actual brain waves. An EEG stands for an electroencephalogram. You may be familiar with an EKG, which studies heart waves. In an EKG, they put little sticky things all over your chest, and then they can measure heart waves. In the EEG, they can put little sticky things all over your scalp, and then they can measure brain waves. We actually will look at brain waves a little bit more towards the end of the chapter as we talk about sleep and dreaming. Another way to study the brain is to actually lesion it. So lesioning is when you destroy on purpose a part of the brain and then study the effects afterwards. There's a couple different ways you could actually cut something out. That's a lobotomy, when they cut out a portion of the brain, or usually, or more commonly, lesioning is where they destroy it. They may insert an electrode into the brain and then they burn it. An example of that would be this rat back here, who he had part of his hypothalamus lesioned. So while the hypothalamus can trigger hunger, it can also tell you when you're full. So they destroyed the side of the hypothalamus that tells you when you're full. And as you can tell, without that part of his hypothalamus, he kept eating and eating and eating, and he got really, really obese. Today, so much has been learned about the brain because of all of the different available neuroimaging techniques. We really just have a small portion of neuroimaging that we really talk about, and I would say you don't have to know what these acronyms stand for, but generally be able to recognize how these work. The first is a PET scan. The PET scan you can usually recognize because it's the most colorful of them. It's also usually a live image. It looks kind of like a radar because it's showing which areas of the brain or the body are most active while you're performing a task. So a person would ingest or drink or swallow or be injected with a radioactive glucose, and then that will show up while they're getting scanned. That's a PET scan. In an MRI scan, it's magnetic resonance imaging. This is best for looking at actual structures in the brain. So it's able to see bones, but it can also see tissues. So seeing structures, the anatomy. Also, if they were looking for a tumor, the MRI would be the best one for that. Now, these machines all do look fairly simple, especially in the way the person is placed into it, but they do all work a little bit differently, and they're not the same machine for all of the tasks. Each type of scan would require a different type of machine. Our next one, the fMRI, or the functional MRI, kind of a combo between the PET scan and the MRI. This one is going to show us blood flow during a brain activity by comparing multiple MRI scans together. During this type, the patient can actually interact with the person who's giving them the test. They can tell them, I want you to think about something sad, or I want you to think about love, or I want you to listen to this story. And then they can see which part of the brains are they using, which parts of the brain get more blood flow to them. That's an fMRI. And then our last type of scan is a CT scan. And a CT scan takes X-ray images of the brain. This is probably the second choice for studying the brain when it comes to MRIs versus CT scans. The MRI is a little bit more detailed because it can see the tissue a little bit better. But the CT scan is usually faster to get an image. So now getting into how the brain actually functions. There are some people who will say that people actually have two brains in their head. And while we don't really have two brains, we have two hemispheres or two halves of the brain that can act very independently of each other. And they're actually very much mirror images of each other. They look, especially on the outside, very similar. They're only connected by the corpus callosum, which is a band of nerve fibers that connect to the two halves and help them to communicate with each other. Your brain, my brain too, practices what's called contralateral control. Contra, the prefix, means opposite. So this means that each half of the brain, that's each side, lateral is side, controls the other half of the body. So the left brain, the left hemisphere of the brain, controls the right side of the body, and the right brain controls the left side of the body. You may have heard that prefix contra before. The place I've heard it is at football games. They talk about that they're going to practice contra flow traffic, which means that all the lanes are going to go the opposite way just to get everybody out of the parking lot the fastest. So in addition to contralateral control, our brain also uses lateralization. That means that each side is specialized in its functions. So they have different jobs. The left hemisphere mostly focuses on verbal processes, so language, speech, reading, and writing. And the right hemisphere mostly focuses on nonverbal processes, spatial relations, including music, and then recognizing faces. So here we have emphasis on this contralateral control. Left visual field to the right brain, right visual field to the left brain, and the corpus callosum are the nerves connecting them. So let's look at the examples over here. We have an apple in our right visual field. Now this is a little confusing because when you're looking at objects, usually you're using both of your eyes, which means that object is actually going to go to both sides of your brain. But let's imagine that you are only looking at this apple with your left eye. So if you're looking at it with your left eye, let me take that back. Let's say we're looking at it with our right eye. If our right eye sees it in our right visual field, then that object is going to follow the blue pathway here. So it's in the blue. Behind your eyeballs is what's called the optic chiasm, and it's going to crisscross to the other side of the brain. So now that apple that was in our right visual field has ended up on the left side of our brain. Now, speech, language, we already said, is controlled by the left side of the brain. So because we saw that apple with our right eye, or even if we were holding it in our right hand, we could say apple. We could say that we see an apple. Now, if we're looking at the pencil with our left eye, the pencil image is going to travel, it's going to crisscross at that optic chiasm, and it's going to end up in our right visual field. Now, in order for us to be able to verbally say that we can see a pencil, that information has to cross to the other side of the brain because that's where language is. So that information is going to cross the corpus callosum to where it'll be in the left side of the brain, and we can verbally say that is a pencil. So a little bit more about the specialization. We don't have to know all of the things that are on here for what the left hemisphere and the right hemisphere does. Generally, again, we want to remember that the left side is language and the right side is spatial and nonverbal. At the back of our brain, we do visual processing, so being able to see words and letters on the left side, seeing geometric patterns, faces, emotional expressions on the right. Hearing language sounds on the left, non-language and music on the right. Memory, verbal memory on the left, nonverbal memory on the right. All of these things related to speech and language on the left, emotional tone on the right. And then spatial abilities primarily concentrated on the right side there. Again, really, really want to remember, left side, language. We used to always see images like this, and if you go on Amazon, you get t-shirts and bags that have this kind of design on it. But that idea of being left brain and right brain dominant goes along with brain lateralization because the brain is better at processing some tasks from each side. However, we know from the myths that the idea of being left or right brain dominant is a myth and that we can actually do and use equally both sides of our brain. All right, our brain also has the special ability to modify and reorganize itself after it's been damaged. So this is called brain plasticity or neuroplasticity. And the younger a person is, the more neuroplasticity they have. So if a young person suffers from a stroke is a blood clot in the brain, they're usually going to be able to recover from that much faster than if an adult or an elderly person has brain damage. So what happens is those areas of the brain that are damaged, the rest of the brain will kind of compensate for that, and they will reorganize those neural connections to form new connections and make up for the damaged area. But again, the younger you are, the more plasticity you have. Talking more about the corpus callosum, this is one of our specific brain structures that we need to know. The corpus callosum, again, these are the fibers that are connecting the two halves of the brain. So imagine that these neural fibers, these are all tons and tons of axons that are grouping together here to form nerves. Now, this view on the right is looking down on the brain from above. The top half of the brain has been cut away so that we can look down here into the brain. And you can see that there's these two halves, and then there's these nerves, all these nerves here that are connecting them. That together is the corpus callosum. If we want to look at a cross section of the brain, the corpus callosum forms this sort of arched piece here. So it is connecting the two halves of the brain. Now, the two halves of the brain are so independent of each other that it's actually possible to cut the corpus callosum, like completely sever it, and have two independently functioning brains. This is rare, but it is possible. And this is usually only done for people who have severe epilepsy or they have severe epileptic seizures. When a person has a seizure, there is uncontrolled neural firing. Their neurons are firing in a way that they're not supposed to. And that firing, the bigger the seizure is, the more firing it is across the brain. So for people who have really bad seizures, that neural firing goes all across their brain. But by cutting the corpus callosum, it limits the seizures to just one half of the brain, that they won't be able to travel to the other side of the brain. That's called a split brain surgery. Gazzaniga and Sperry are the two men who are most famous for this, and I think they might have even won a Nobel Prize from doing this. But they found some quirks that happened after these people had these surgeries. They didn't mess with their brainstem, the lower part of the brain, so they were able to still live and function. But because of that contralateral control that they didn't have anymore, there were some weird quirks. And here's one of them. The split brain patients were asked to focus on a dot, and then they were shown things in their left and right visual field. So things were just on the left or just on the right. When they would show them something in the right visual field, they could name that object and they could describe it. Because remember, if something was in the right visual field, it's going to go to your left brain, and the left brain is where language is. But if they showed them something in the left visual field, they couldn't actually say it. They kind of knew it was there. They could actually draw it, but they could not verbally say it because that image was stuck in the right side of their brain and it could not cross to the left side without a corpus callosum. So it supports the idea that language is controlled by the left side of the brain, that language really is just in that one side. Pretty neat. All right, that is it for flipped notes 2-4.